the thing that really strikes me is that been really the most comprehensive view that we've ever had of the response of the human body to spaceflight. So, Mark Kelly stayed on the ground for the year, and while Scott Kelly was on the International Space Station for a year, and we were able to monitor both of them before, during, and after the flight. So we could then, by all of these different endpoints that we were looking at, we could attribute space flight to being the effector, what was causing these things, and not just genetics differences between the two because they're the same. This was definitely the first time that NASA's ever done any of the kind of omics studies. So you think about genomics where they sequence the DNA, or they transcriptomics, they sequenced the RNA, they looked at the products of metabolism, metabolomics. They looked at changes in proteins. They really did a lot of those types of studies, cutting edge kinds of science. Our study then looked at, monitored or followed Mark and Scott's telomere length before, during, and after the flight. And what we saw actually was exactly the opposite of what we imagined, because we imagined all the stresses and exposures of spaceflight would act to shorten Scott's telomeres in flight. But in fact, he had longer telomeres in flight. So every single sample, every single time point we had in flight, his average telomere length was longer than it was before he went up. So that was really a surprise to us. The twin study will really guide a lot of those future kinds of studies. And that's, again, another one of the really important things about it is it's laid the foundation. It's a landmark. We set the baseline, and so we know what to watch for going forward.